Good afternoon, I guess. Good morning for some people still. So my name is Oliver Rosen. I'm the Chief Medical Officer of SQZ Biotechnologies, and I'd like to welcome you today for this webinar on anal cancer. What is important to mention is that we meet with the patient foundations that are active in diseases that are driven by HPV-16 positive cancers. And it turned out that kind of the needs are similar across the different diseases. The challenges are very similar, very similar, and so that's why we decided really that we do a lot of things together with all different foundations. But obviously today is catered to anal cancer, and the objective really of this webinar is first and foremost to understand really the patient perspective, somebody who is diagnosed with anal cancer, and then the treatment options from a medical oncology perspective, and also the importance of radiation therapy in these areas. And we will talk about the first clinical trial that is run by SQZ Biotechnologies. And so I'm really honored to welcome our panel here. And as you can tell, we're all socially distant, just for the record, 6 feet apart at least on our screens. So we have Diane Jones, who is, will describe the patient journey. And Diane is an ardent patient advocate. She's a herbalist, holistic health coach, a writer, and an educator. And then we have Dr. Kathy Yang from Vanderbilt Medical Center. She is the gastrointestinal cancer research lead for the gastrointestinal cancer research program, professor of medicine for hematology and oncology, and the co-director of the GI program at the Vanderbilt Ingram Cancer Center in Nashville, Tennessee. Then we have delighted to have Dr. Emma Holliday, assistant professor in gastroenterology, radiation oncology section at MD Anderson in Houston, Texas, and Dr. Howard Bernstein, who is our chief scientific officer at SQZ Biotechnologies. And so, Diane, thank you again for your participation. We'll hand it over to you now, please.
Wonderful, thank you. Hello, everyone. My name is Diane Jones, and I was diagnosed with squamous cell anal cancer about 5 years ago, 5.5 years ago. And some of the classic symptoms of anal cancer, as you can see from my slide, are anemia, rectal bleeding, anal itching, straining during defecation, narrow stools, or other changes to bowel habits, including fecal incontinence, black tarry stools, fatigue or exhaustion, a lump or mass at the anal opening, an abnormal discharge from the anus, pain or sensation of fullness in the rectal area, swollen lymph nodes at the groin or in the anal area. Some of these symptoms you see have an asterisk attached to them, and these are the symptoms that I dealt with, but never all at once. So, you know, I had one bout of rectal bleeding. I had one bout of anal itching. Didn't realize that I was anemic until we did a blood panel. So how did I end up being diagnosed? I could not donate blood because of the anemia. Went to a doctor about that, and he recommended that I have my colonoscopy. So it's important to understand that you don't get all of the symptoms all at the same time, and you should check everything, okay? An iron panel did reveal my anemia. Next slide, please. Thank you. So I went and had a colonoscopy and an endoscopy and a biopsy. And my doctors at Memorial Sloan Kettering called to confirm a diagnosis of squamous cell anal carcinoma in April of 2015.
Oh, God.
I was given a mitomycin. I was given the standard care of treatment, mitomycin push on the first day of treatment, and I was supposed to have one on the last day. Capecitabine oral therapy twice a day, once in the morning, once at night, on the days when I was having my radiation therapy. my radiation therapy every weekday with holidays and weekends off. And in my case, I was a problem child, as my doctor called me, and I did not respond well to the capecitabine. It was very, very toxic to me. So I ended up hospitalized a few times. And in my case, in the final week of treatment, I ended up with a 96-hour 5-FU infusion. Okay, and the capecitabine was discontinued, and I did not get my final mitomycin push. My team at Memorial Sloan Kettering included my surgical oncologist, my radiation oncologist, my medical oncologist, and gynecological oncologist. And I was initially followed up by various members of my team once every 6 months, then once annually. Of course, during treatment, I saw my doctors a lot more regularly. Okay. Next slide, please. Thank you. As Oliver said, I am an advocate. I advocate for anal cancer. I advocate for breast cancer. A family member was diagnosed with breast cancer in 2009. When that happened, I immediately signed up for the Susan G. Komen 3-day walk to end breast cancer, walking 60 miles over a 3-day period. And I've participated in that every year, including the year that I was diagnosed. My doctors actually gave me approval. So this relative's cancer metastasized in 2013, so less than 5 years later. She was enrolled in a clinical trial, and I have many friends and family members who have been in clinical trials. They're very important to me. So with my own diagnosis, I wanted to share my personal story and improve awareness surrounding anal cancer. How did I do that? Where did I go? When I tried to give blood, New York Blood Center turned me down, and they didn't have a platform to share my story, even though they tried. Memorial Sloan Kettering has many wonderful programs to help patients, but they don't allow you to become an advocate until you're a full year out of treatment. And I stumbled upon the HPV Anal Cancer Foundation peer-to-peer program. I found them in 2016. I've been with them ever since. I have been a peer match for, I believe I'm up to my 16th peer match. So it's been a very rewarding and important personal experience for me, okay? And I offer all of you who are dealing with anal cancer to look into the HPV Anal Cancer Foundation and see if you can get some help, okay? Next slide, please. Some of the things that patients go through when they are diagnosed with HPV, anal cancer, or anything like that, is pretty much, there's stigma attached to it. There's a huge stigma attached to it. Not only stigma around having an HPV cancer, but around the very name. Anal cancer is very descriptive. And when you see it, when you say it, you get to hear and see people flinch. One of my family members said, when we tell people, can't we call it colon cancer instead of anal cancer? And my answer was no, we have to call it by its appropriate name. One of the things I've learned through my advocacy is that all HPV cancers do have a stigma attached to them, even though one can develop an HPV cancer with just one sexual partner. It's not a moral issue. In some communities, patients with HPV cancers are made to feel ashamed, judged, and are sometimes shunned. And one of my peers actually told me that she, when she was newly diagnosed, had to deal with that emotional baggage in addition to the hardship of treatment, in addition to the emotional baggage of just being diagnosed with cancer, in addition to all of the pain and suffering that goes along with a cancer diagnosis. And I've been a caregiver and I've been a patient, and I will tell you that I think that being a patient is easier. So this stigma is in addition to the burden of treatment and caregiving. And
Diane, I just want to interrupt for 1 second. Galaxy A20, participant Galaxy A20, we can't mute you centrally. Could you please put yourself on mute? We want to be respectful to Diane's presentation, please. Yeah, Galaxy A20, we cannot mute you. You're calling in from a phone. So please put yourself on mute. Thank you very much. Sorry, Diane.
That's quite all right, Oliver. Thank you. Okay, so dealing with the burdens and pain of a cancer diagnosis is difficult enough, but then sometimes you have a reoccurrence. Next slide, please. Okay, as I said, at less than 5 years after my relative was initially diagnosed with breast cancer, it metastasized. She was given a poor prognosis. We were actually told that we could end things within 2 years, within 2 months, and she was given possibly.
I'm sorry, Galaxy A20, could you please put yourself on mute? We want to be respectful with Diane when she shares her story. So please put yourself on mute. We can control everybody else, but we cannot control you. So please put yourself on mute. No, I wish we could do this here. So thank you for that hint, but we cannot because the person is calling in from a cell phone, I guess. So Diane, I sincerely apologize.
No worries. Thank you, Oliver. Okay, so this relative was told that she had between 2 months and potentially 3 years to live. She was enrolled in a clinical trial more than 7 years ago, and she's thriving today with her diagnosis of metastatic breast cancer. So what are some of the benefits of clinical trials in addition to potential life-saving? Clinical trials help in discovering efficacy, safety, and tolerance of new treatments. And the current standards of care that we are using right now are all thanks to the willingness of patients who have come before and been willing to participate in clinical trials. Okay, next slide, please. Thank you. The benefits of the SQZ Biotechnologies approach is twofold, and everyone here will explain further, but the therapy is personalized to the individual using their own blood. It's not an overarching kind of clinical trial. And it is believed that this therapy, by squeezing antigens into blood cells, will be better tolerated by the patient. Next slide, please. Okay, and I don't know if you want me to describe all of the terminology that might be used in the rest of the webinar, but I did put together 2 slides explaining some of the terminology, and I've underlined things like PBMC, peripheral blood mononuclear cell. That's any whole cell having a round nucleus, and these cells consist of lymphocytes. Those would be your T cells, your beta cells, and your natural killer cells, as well as monocytes. So you can go ahead and look at those during the course of the webinar if necessary. And I think I will end there.
Diane, thank you so much. And again, I'm sorry for the disturbance, but I guess everybody's just focused on your talk because it's obviously, you pick everybody up and very kind of engaging. And so just the last slide here just shows what an amazing educator you are. So thank you for participating and really supporting the Anal Cancer Foundation with your efforts. So I think now, and what I should also say, and I apologize if I didn't say this before, we'll do questions and answers at the end. So please keep your questions. Some questions might be addressed during the talks of others. And so I might now invite Dr. Kathy Ankleys to take it over.
I'm waiting for you to turn my video on.
Done.
Thank you. It's my pleasure to present today. Basically, I'm giving all of you a background regarding anal carcinoma. Thank you once again for inviting me to speak on this webinar. I hope it helps several individuals. I'm glad to see that the SQZ Foundation is also involved, as well as advocacy. Next slide. So just a reminder, anal carcinoma has been rising in incidence over the past almost 2 decades. It's been increasing by 2.2% per year. Next slide. And the average age patient is 62 years old. The average person is a Caucasian female. And the younger patient population may be the more HIV positive or immunosuppressed patient population, just to keep that in mind as well. The majority of patients do present with more earlier stage disease, meaning that they don't have metastatic disease. Metastatic disease at presentation is not common. It represents only 10 to 20% of all of the patients originally diagnosed. Next slide. So next slide. I'm going to just be talking a little bit about locally advanced disease. We have Emma Holliday on, who's our amazing radiation oncologist, but I think as Diane mentioned, the standard of care traditionally has been 5-FU and mitomycin 2. The reason is because this is found to be basically curative in the sense that you could avoid surgery. It has been compared to 5-FU cisplatin in a former trial, although that trial was a bit complex because it also included providing chemotherapy up front first before proceeding with tumor radiation therapy. And at the end of the day, basically, the new regimen did not demonstrate to be superior to 5-FU and mitomycin. Next slide. And the same is true for the ACT-2 trial, which is the largest study ever conducted and which is a true head-to-head comparison. Next slide. And what that study demonstrated was, in fact, that they are equivalent in regards to outcomes, just differences in toxicities. But once again, it did not demonstrate any superiority for the new 5-FU cisplatin consideration of a newer regimen versus 5-FU mitomycin. That is why 5-FU mitomycin still is considered a standard of care, because nothing has been better than the 5-FU mitomycin. But in fact, 5-FU cisplatin is equivalent based on the study. Next slide. So what are some new approaches to earlier stage disease? Next slide. Want to highlight this trial that's just opened, 2182, focusing on reducing the intensification of chemoradiation therapy and hopefully reducing toxicities. And this is being led by Jennifer Dorff, who's a radiation oncologist. Next slide. And it's specifically for very early stage tumors, so T1 or T2 tumors, so small tumors, less than 2 centimeters with no lymph node involvement. They're randomized in a 2-to-1 randomization to the investigational arm, which is once again with a reduced dose of radiation therapy. And the premise for this is because the original trials going back 3 decades ago actually provided lower doses of radiation therapy, and they were still found to be effective. So the thought process is, can we go back to the lower doses of radiation therapy and reduce the toxicities for earlier stage disease? And so this trial is ongoing, and please, if you know of an individual with very early stage small tumors without lymph node involvement, please consider enrollment. Next slide. So what about metastatic anal carcinoma? I think many of you that may have followed my research path, that has really been the premise of a lot of the work that I've conducted, and that is in large part because there was no standard of care for this patient population. Next slide. And historically, basically, while a few years ago, several years ago actually, we had conducted one of the largest studies looking at standard chemotherapy regimens and trying to identify which chemotherapy regimens would be ideal for patients, because there had never been a randomized trial provided to newly diagnosed patients. Next slide. And more recently, however, the first randomized phase 2 study has just been recently reported, which I was very glad to participate in and served as a USPI for this study. And this is actually based upon a lot of the work we conducted and completed while I was at MD Anderson, and served as the premise for this trial, comparing 5-FU cisplatin versus carboplatin and Taxol for newly diagnosed metastatic carcinoma patients. Primary endpoint here was response. And if response was equivalent, then we were going to determine which regimen had less toxicity. Next slide. Now, this is an international trial. And what we found out that in fact for newly diagnosed patients, the response rate was quite high, close to 60%. Next slide. And the progression-free survival, as well as the overall survival, appears to be in favor of the carboplatin and weekly paclitaxel arm. It should also be noted that the toxicities were actually greater in the 5-FU and cisplatin arm, keeping in mind this is a very traditional schedule of once a month 5-FU and cisplatin, which is a higher dose of treatment, which means that obviously there's higher potential for toxicities. Next slide. Now, what about immunotherapy? As many of you know, that is now has a large role in several malignancies. Next slide. And we know that greater than 90% of all squamous cell carcinomas of the anal canal are associated with HPV, or human papillomavirus. So as a result, this was a trial that actually completed. It was a multi-institutional study that we conducted and was published in 2017, looking at patients that had received at least one prior line of chemotherapy, and basically allowed them to receive single-agent nivolumab as on a Q2-week or every 2-week schedule, with the primary endpoint being response. Next slide. And these were the results that we had. Basically, we had a response rate of about 24%, and some of you may say, well, that doesn't seem to be such a great response, but in fact, that's a very good response, especially if you're talking about reducing tumor growth. We're not including the patients with stable disease in that number. These are patients that had at least a reduction that is noticeable, as well as these were heavily pretreated patients, meaning they had had at least a median of 2 prior lines of therapy even before going on this trial. And time to tumor progression or progression-free survival was 4.1 months. The median overall survival was 11.5 months for this trial, and we had very few toxicities on this study. Next slide. And this is an example of what we were seeing with our patient population. This, in fact, is one of my own patients that was on treatment for almost a year. Very, very few side effects, had a very good, basically, quality of life. She felt significantly better while on treatment. Next slide. As you also know, there's also pembrolizumab, which has been created by Merck. They had their phase 2 study just recently reported, basically same concept, looking at patients that had received prior therapy. They, however, required patients to have, originally they looked at patients with high PD-L1 expression versus those that did not, and the primary endpoint was also response. Next slide. This was just reported earlier this year, and they noted, in fact, that the progression-free survival was 2 months, and the overall survival was 11.9 months. The response rate was about 10%, so a little bit less than what we reported. The study was about 100 patients. Next slide. Now, what about ongoing trials? Well, you should be aware that because of our work with nivolumab in the NCI-sponsored study, and because of pembrolizumab from the work from Merck, the NCCN treatment guidelines have approved those regimens as a possible option for patients with previously treated disease. However, there are additional trials that are ongoing that you should keep in mind. These are not FDA-approved treatments because these were not written specifically for FDA approval, and honestly, the FDA does not take this into account because it's considered a rare cancer. So EA2165 is a trial looking at for a high-risk patient population that basically, if your tumor is greater than 4 centimeters, if you have no positive disease after completing chemoradiation therapy, you can be randomized to 6 months of nivolumab for standard surveillance. This is an ongoing trial, and basically is enrolling quite well and is almost near completion. Next slide. And then to expand further on the original immunotherapy trial we created, this trial is about 2/3 finished with enrollment. This is NCI Part 2. Basically, not only, once again, 1 prior line of therapy, patients are now randomized to an easier schedule of nivolumab, only once a month, not every 2 weeks, plus or minus ipilimumab. This trial is currently ongoing, and you should keep in mind, each of all the new trials do allow HIV-positive patients, as long as the CD4 count, usually for the most part, is greater than 200 or 300. So once again, this trial is almost, it's about 2/3, a little bit over 2/3 filled with enrollment. It's a multi-institutional study as well. Next slide. And now we are very proud to have a phase 3 trial. It's the first time ever in newly diagnosed metastatic anal carcinoma patients based upon the 2 trials I just showed you. Next slide. And it literally just opened about less than a month ago. It's a 2-to-1 randomization of carboplatin weekly paclitaxel based upon the work of Interact, which you had seen earlier, plus or minus nivolumab with the continuation of nivolumab for maintenance therapy. And the primary endpoint is progression-free survival. So we hope to improve the time to tumor growth by an additional roughly 3 to 4 months with the investigational arm. So this trial literally just opened up. It's the first NCI-sponsored trial, and it's open throughout many of the neighboring hospitals and academic institutions as well as community practice. Next slide. And I believe now I'm going to hand it back over to you, Oliver. Thank you,
Kathy.
And thank you once again to all the patients. And I would like to emphasize the same as Diane. Participating in clinical trials is critical because we cannot move treatment advances forward unless you participate in a clinical trial. If patients had not participated in our earlier clinical trials for metastatic disease, this would not have resulted in the improvement and development of new trials for metastatic disease as well as earlier stage disease. So thank you so much for your attention.
Thank you again, Kathy. So I'd like to hand it over now to Emma Ute, please, and I will change here the video formatting. And so 48, please, slide 48, please.
Thank you so much for the opportunity to speak today. My goal will be to give an overview on the role of radiation therapy in the treatment of anal cancer, particularly local regionally confined or non-metastatic disease. Next slide, please. So I wanted to start with a brief history of the treatment of anal cancer, non-metastatic anal cancer. And it started 40 or 50 years ago. At that time, anal cancer was primarily a surgical disease. It was treated with a radical surgery called an abdominal perineal resection, and this left the patient with the permanent ostomy bag, which led to a severe impact on patient quality of life. Not only that, but surgeons found that recurrence rates, even after such a radical surgery, were pretty high. About 50% of patients had their tumors come back in the pelvis, even after the abdominal perineal resection. Because the patients and the surgeons and the medical community weren't happy with this high recurrence rate, a surgeon named Dr. from Wayne State started off with a small trial treating with a low dose of radiation before his operation to see if he could shrink the tumor down and make his operation more successful. This is what we were doing at the time in rectal cancer, and it seemed like a reasonable way to insert radiation to help the surgeons out. What he found, however, was very interesting and surprising to everybody. He found that the majority of patients in his small initial trial actually had no live tumor when he went to do his operation. So this started the paradigm shift, and it started a new way of thinking that perhaps locally confined disease, anal cancer, could be treated with chemoradiation alone. Over the next several decades, there were more trials trying to improve on Dr. Nigro's regimen because he essentially pulled it out of thin air. He extrapolated from how we treated other cancers at the time. It wasn't empirically derived. So there was a cascade of other trials that followed Dr. Nigro's original work to try to improve on that initial regimen. His initial regimen included 2 chemo drugs, 5-FU and mitomycin C, and a pretty moderate dose of radiation to the anal tumor as well as the lymph nodes in the pelvis. Even after all these trials, long story short, nothing was able to improve upon this original recipe. They tried dropping the chemo altogether. They tried dropping one of the chemo agents. They tried swapping the cisplatin for mitomycin C, as Dr. Ng mentioned in her talk. None of these were shown to be superior to that initial regimen. So that remains the standard of care. Next slide, please. So to summarize, essentially 50 years of research in non-metastatic anal cancer not much has changed. We still use that same regimen of daily radiation for anywhere between 5 to 6 weeks, Monday through Friday. We give 2 chemo drugs concurrently with the radiation. The standard of care for most doctors throughout the country is still that mitomycin C and 5-FU. There are other chemo regimens that are considered acceptable per the national guidelines. As Diane mentioned, some doctors will swap capecitabine, an oral pill, for the 5-FU. Some doctors will switch the cisplatin for the mitomycin C because although it wasn't better, as Dr. Ng mentioned, it certainly wasn't worse and maybe less toxic. So that's where we are today. Not a whole lot has changed. We have a few more options, but in general, we still use radiation as the primary driver to cure non-metastatic anal cancer, and we give that radiation with chemotherapy. So, you know, what patients most often ask me first when I'm meeting with them in clinic, you know, as we've mentioned before, anal cancer is a very rare disease. Most people don't know somebody who's been through treatment or who's been through this journey. So a common question we get in clinic is, what are my chances? You know, how is this curable? How am I going to get through to the other side? And, you know, there are prognosis numbers and percentages that were published from one of the major trials. I put it here on this slide, RTOG 9811. And, you know, you can see that there's quite a big range. For people with earlier disease, smaller tumors with no lymph nodes, you know, the survival rates are quite good, between 80 and 90% at 5 years. But then as you go further down that list, and if you click, a little box should pop up. You know, if the tumors are large, particularly if the tumors are large and there are positive lymph nodes, you know, we see that the survival numbers are really not where we would like them to be. Next slide. So, you know, we can see from the prognosis or the treatment failure numbers I showed you on the last slide, you know, what we can expect with these standard doses of radiation that we've been giving for several decades and the standard chemo that goes along with it, that just isn't enough for some patients with more advanced disease. So another common question I get from patients sitting with me in clinic with this new diagnosis is, why can't we just dial up the dose of radiation? You know, you're giving me radiation every day, just turn up that dial, give a little bit more. We should be able to give enough to burn up that tumor. And the reason we can't do that is we are humbled by the toxicities or the side effects that radiation gives when we direct it at the pelvis. I like to tell my patients that the pelvis is very expensive real estate. There are a lot of important organs that are near the anal canal and the lower part of the rectum. And while radiation is very effective at killing squamous cell tumors in the anal canal, it also comes with high rates of side effects that not only impact how patients feel during treatment, but can lead to scar tissue formation, fibrosis, and problems with how the organs in the pelvis function long term. So, and if you click, we should have a little box come up. There we go. So, you know, one way we as a radiation oncology community wanted to try to improve the side effect profile, as this treatment we know is very effective, is to use a more advanced technique to deliver that radiation. So most of the trials you've heard about today used an older technique where we aim some beams straight on, one from the back and a couple from the sides. It's you know, fairly simple in how the radiation is delivered. There was a group of doctors kind of in the early 2000s that wanted to utilize a newer form, a newer technology of delivering radiation in order to target the radiation being better at the tumor itself and try to minimize unnecessary dose to the other organs in the pelvis. And this technology is called IMRT, or intensity modulated radiation therapy. And you can see from these two pictures I put on this slide, you know, the one on the left uses those 4 beams, one coming in from each side. And the dose distribution for the picture on the right is given using IMRT. So we use more beam angles, the energy of the beam is modulated, and essentially the radiation dose is pinpointed better at the tumor and at the lymph node areas. So we can avoid dose to things like the bowel, like the bladder, like the vagina or the male genitalia, and also unnecessary dose to the skin to minimize some of those radiation skin burns that can be so painful. Next slide, please. So after all of that, how do I treat patients today in 2020? First of all, I do use that more conformal pinpoint type of radiation called IMRT. There's a cousin technology called VMAT, which stands for volume modulated arc therapy. Those can sometimes be used interchangeably. And those, as I said on my previous slide, allow us to really target the radiation dose better and to reduce the risks of side effects compared to the older techniques. Additionally, I customize the radiation dose for patients depending on the size of their tumor. We know that smaller tumors are easier to kill, easier to cure, and they may not need quite as heroic a dose of radiation therapy. This allows us to minimize the side effects for patients who don't need that higher dose of radiation. For patients who do have the larger tumors or who have lots of lymph nodes involved in their pelvis, we dial up the dose a little bit. For reference, we go from somewhere around 50 to somewhere around 58 gray, which is our unit of radiation. And what we've seen in some of the smaller trials we've done at our own institution is that patients do seem to do better when the larger tumors receive a larger dose. Next slide, please. So after we talk about what are my chances, doc? What is treatment going to look like? How are you going to deliver it? The next set of questions I get from patients in my clinic is, how am I going to feel? Am I going to be able to go to work? Am I going to be able to take care of my kids or grandkids? And a lot of my patient visits, a lot of time is spent in describing the toxicities or the side effects that they're going to feel during radiation. And, in general, we divide them up into acute and late toxicities, short-term and long-term. And the short-term toxicities are pretty much guaranteed. Almost everybody experiences them to some extent. They get radiation dermatitis, which is the $10 medical word we give for a skin burn. Usually this is worse around the anus itself and in the area of the groin. In terms of genitourinary effects, patients will sometimes experience frequent or painful urination. Men can have obstruction of their urinary flow if their prostate swells during radiation treatment. A combination of chemotherapy and radiation both will lead to diarrhea, which almost always needs to be managed with either one or two medications. And more than that, patients can have the sensation like they have to move their bowels all the time. We call it tenesmus or spasming of the low rectum and the anal canal. And this can be pretty alarming and bothersome if people are having the sensation constantly throughout the day that they've got to move their bowels. Other side effects that don't show up on as many forms or sometimes aren't discussed as much as I think they should be are pain and fatigue. The anal canal is a very sensitive part of the body, and causing what's akin to a sunburn where the sun don't shine is extremely painful. So I warn my patients that they will very likely, almost certainly, need prescription pain medicines to get through, especially the last part of treatment. And also, radiation just tends to make you feel really tired. It tends to wipe you out, kind of sap you of energy. Next slide, please. Once I give patients an idea of what to expect during treatment, I also like to give them an idea of what long-term side effects can be caused by radiation to the pelvis. And these are things that don't happen to everybody. Thankfully, the percentage of severe long-term toxicities is pretty low. But we do know that most patients will have some change from their baseline function after radiation to the pelvis. The most common manifestation that my patients tell me about is that their bowels just never quite work the same after radiation for anal cancer. Whereas they may have moved their bowels once a day before, now they're going a couple times a day. Their bowel movements may be more urgent. They may be more sensitive to certain foods that trigger diarrhea. And the spectrum of how much it bothers folks has a pretty wide variety. So some folks are very bothered by these, and some folks, they're able to adjust just fine. There can also be scar tissue that form in other organs in the pelvis that can lead to some of these other late toxicities, the bladder, and the healthy small bowel. Sexual dysfunction is also, I think, an under-discussed and under-warned about toxicity profile from radiation to the pelvis. Next slide. So this is my final slide, and Dr. Ng mentioned both of these trials in her talk, so I'm not going to belabor them here, but I did want to give you an idea of where we're going and how we think we can do better for patients with non-metastatic anal cancer. There's essentially two groups of patients. There's one group of patients that are very early stage, small tumors, no lymph nodes, and we think we can do better for them by maintaining their excellent cure rates and dialing back treatment a little bit to reduce the risk of some of these long-term quality of life limiting side effects from radiation. That's the decreased trial that Dr. Ng mentioned. The other group of patients are folks who have larger tumors, that started to spread to the lymph nodes. And for those patients, we really aren't where we want to be in terms of our cure rates. And so for these patients, we really need to be thinking of ways to turn up the dial. Maybe we can't turn up the dial in terms of dose of radiation much more, but we can think about creative ways to sequence or combine other therapies to improve the cure rates for this particular population. So that's where we are going. That's where we want to go in terms of the treatment of non-metastatic anal cancer. It's really about individualizing therapy and not using the same one-size-fits-all approach that we've been doing since 1970. So thank you for your time. I understand we'll take questions at the end, and I look forward to the future talks and further discussion. Thank you.
Thank you so much, Emma. So 2 slides forward, please. So Howard, I would ask you now to be brief, and Veronica, please go to slide number 40 now, please. 40, yes, and then we jump to this one.
So good afternoon, everyone. Good afternoon, and thank you for participating in this webinar. I'll spend just a few minutes telling you how we make our treatment and how it works. Our treatment is what you would call a cancer vaccine, where we take your own blood cells and we process them and we train your cells to stimulate your immune system to produce another type of immune cell that actually will kill the tumor. So basically, as shown on the bottom, we take out your cells, we put them through our cell squeeze technology, we create the vaccine, we then inject it back into you. Your cells go to your lymph nodes and your spleen, where they stimulate what are called T cells, which have the ability to kill tumors. Next slide, please. Typically, the way this will work, the subject goes to their physician, where your blood will be removed. This is day 0. We then take your blood and we ship it overnight to our manufacturing facility. We then process the blood using our squeeze technology, and I'll spend a minute showing you how that works. It takes about 17 to 18 hours to produce the vaccine. We then put the vaccine through strict quality control testing to make sure the vaccine looks appropriate for use. And then we ship it back to your physician approximately a week later. And the therapy is then administered via syringe into one of your veins. And the nice thing about this is no preconditioning is required prior to using the vaccine. Next slide. So the way we work, we have a technology which is called squeeze. We have these tiny little channels that we push cells through. And as they move through these channels, there's a constriction which actually squeezes the cells. And in the process of squeezing the cells, we take materials, synthetic materials, so we chemically synthesize these. These materials look like materials in your tumor. When we squeeze them, these materials go into these immune cells and activate the immune cells. And so this is how we produce the vaccine. It then goes for quality control testing, and it's ready for use. Next slide, please. And then the vaccine, and again, is administered intravenously, and the cells go both to the lymph nodes and to your spleen, which is also another lymph organ. And here I show, if you were to look inside these organs, at various time points. On day one, you can sort of see something is happening in these organs, and that's where we're starting to produce the T cells that can act against the tumor. By day two, you can see the colors. There are many more of these T cells. And then by day three, we have a significant number of T cells. After the third day, these T cells then leave these organs and migrate into the tumor. Next slide.
Which is slide 62, please. So just flip through this quickly.
And here are some studies that we have done in animals, and where we've implanted tumors in the animals. And so we're showing tumor growth over time. And what we see, if you look on the top of the graph, we vaccinate the animals at approximately day 14. The immune system is then making the T cells which kill the tumor cells. That's the second arrow. That's approximately 3 days. These cells then leave the tumor, the lymph nodes and spleen, and then migrate into the tumor. And then shortly after, you can sort of see the tumor stop growing and start declining. And there's a period of time which is common with immunotherapies, where T cells will migrate into the tumor. The tumor can still grow while the T cells are there, and it takes a while for the activity of the T cells to kill significant amounts of tumor, so the tumor starts shrinking. So I will now turn it back over to Oliver, and I'd be happy to answer questions later on.
Thank you, Howard. So the next slide, please. Next slide, please. So it came up already in Diane's talk, particularly, and so I think it is important really to spend a minute on the question, why may one participate in a clinical trial? And so all the experts would say there are two reasons to consider a clinical trial. One thing is that one might have interest in getting access to a treatment that might be better than the current standard of care. And the other option might be that your doctor cannot really recommend a good standard option. You might remember that Dr. Eng, for example, said sometimes there were things that were kind of similar. And in this case, a clinical trial might be an option. What is also important is to emphasize that you might not always benefit from a clinical study directly. Sometimes future patients might benefit from a study that might lead to a better safety or a better activity. When you participate in a randomized trial, you would at least receive standard of care. And this was also kind of a notion that Dr. Holliday brought up, is this, we would not be here without all the different tests, all the different trials that we did. And Dr. Eng also obviously asked for participation in clinical trials. So with this now, Veronica, please go to slide number 44, I'm sorry. So it is very important to emphasize when we talk about this cell product that Dr. Bernstein just mentioned, which is called squeezed PBMC HPV, to emphasize that it is in early development. It has not shown that it can effectively treat cancer. It is very important to emphasize this at the beginning. It has been shown so far that it's a safe treatment, but we do not yet know whether it will really help patients. As you all know, currently we have discussions about the COVID-19 vaccines. There's a vigorous process behind until the FDA, for example, would recommend a treatment option. So it is a blood-based cell cancer vaccine, and patients have to be HPV-16 positive, because that's what it's catered to, the treatment. And it might also improve the efficacy of immune therapies. You heard from Dr. Eng, different types of immune checkpoint inhibitors. And so it might be effective in combination with these molecules, and that is also part of the trial. This will eventually be tested also. Next slide, please. So who can participate in the trial? Patients have to be 18 years or older, and one test needs to be done, which is a type of blood group testing, so to say, for white blood cells. They need to be HLA-A2 positive, and then the sites, and we will have at the end a list of participating sites, They will test also and confirm whether your tumor is HPV-16 positive. And then you have to be able to undergo kind of a collection of blood cells, which is called leukapheresis. That takes approximately 2 to 3 hours, and that is done 1 or 2 weeks before the start of the treatment. Next slide, please. I had mentioned that we eventually will test combination treatments. So we have here different combination cohorts that we will test in combination once we identify a very active monotherapy group. And then we might even go to a triple combination, because that's just where kind of the trend is going now. People want to have effective treatment options. So Veronica, if you could go now, please, to slide 67, please. So this is what is called a first-in-human study or a phase 1 study. That's why I mentioned at the beginning, we don't yet know whether it's actively treating cancer. Patients will be treated at different dose levels of the drug, which is called a dose escalation phase. And then there's also an expansion phase planned for monotherapy, and then also the combination cohort that I mentioned, the different cohorts that I mentioned. I spoke already about the HLA-2 positivity, and treatment will be repeated every 3 weeks in this trial. The next slide now, please, Veronica. So what do we measure? One thing is important also in a clinical trial, that sometimes you undergo more tests than you would do for standard of care. But I think both experts talked about the benefits of a clinical trial. And so what do we measure in this particular clinical trial? We measure how often adverse events occur. You heard adverse events that are associated with current treatment options. We call this also safety. And then we test whether patients need to modify or discontinue treatment with this cell product. This is called also tolerability. And then we test what the squeeze PBMC HPV does in your body. This is called a pharmacodynamic effect also. And then we measure how much it reduces the tumor size. And we measure also at which dose level it should be tested in future studies. This is called a recommended phase 2 dose. So if you could go please now to the next slide and final slide. And so this is a slide here which lists all the different participating sites. So Dr. Eng is the principal investigator at the Vanderbilt University Medical Center in Nashville, Tennessee. We also list here additional resources. There's a website called clinicaltrials.gov. Our different advisors from patient foundations mentioned that it's sometimes difficult to understand. So that's why we have on our website also kind of a simplified version for this. And we obviously are always there if you have any questions, if you write at this email address that it was also required for registration. So with this, I would now call everybody for questions. If you have questions, we would like to address them now.